Okay, good afternoon. Yesterday we discussed about some basic aspects of DNA, RNA, uh, protein synthesis, as well as the gene and gene expression. Today we shall discuss another topic, DNA replication and DNA repair mechanism, because these are also associated with fundamentals of your topic, um, gene expression and regulation. Do you have any question from my previous first lecture? If no question, then I shall straight away start the lecture on DNA replication and DNA repair mechanism. Can you see the slide?
Yes, sir.
Okay. So our today's topic is DNA replication and DNA repair mechanism. People thought in the past that DNA is stable, and this is why uh, the organism maintains its properties. For example, human being, uh, offspring of human being, like human being. That means hereditary characters are maintained. But DNA practically, over the years of research, scientists understood that DNA is also prone to decay or damage. But DNA has the mechanism of uh, repair and maintenance of any decay. So today we shall discuss on some fundamental aspects of DNA replication and DNA repair mechanism. DNA, obviously a mystery of life from one cell to another, from one generation to the next. It is, transmitting the hereditary characters. The genetic information that governs how human beings are shaped has flowed through our bodies from hundreds of thousands of years, not only for human, but also for it is applicable for other organisms. And it is constantly subjected to assault from the environment, yet it remains surprisingly intact. Uh, Please be attentive on this statement. It is constantly subjected to assault, means DNA is constantly subjected to assault. Assault means under stress, and also uh, some of the stress can cause damage to the DNA. So from the environment means UV light or many other, materials that can affect the factors can affect the structure of DNA. But it still surprisingly remain intact. It is very interesting. Replication general principles is a start must be ready, uh, must know where to start. So DNA replication is essential. Yesterday I told you, when cell divides, before the division, DNA replication is essential because to transfer the all information, genetic information to the daughter cell. So my start and finish, these are two steps of DNA replication. Must be ready DNA, a cell must be ready to replicate and must know where to start. And finished must all finish, must ensure that each piece of DNA is replicated only once, and there must know where to finish. These are the basic principles. The replication of DNA actually proposed by the Watson and Crick's paper because they noticed that uh, DNA before the cell division must replicate, and coping mechanism of DNA, although they had not enough evidence based on the previous knowledge and understanding about the impact of DNA to the uh, traits of an organism as well as hereditary uh, uh, principles. They propose that DNA must replicate before the cell division, and DNA can make a copy, like photocopy of its own before the cell division. So accuracy and fidelity is really a big problem during making a copy of DNA. Sometimes, uh, it is a big polymer, big molecule nucleic acid. It needs, proofreading or repair if there is a mistake, and must distinguish between original and copy. So these are the, if you make a copy of a big book, you have to do proofreading, and if there is a mistake, you need to edit it. Similarly, DNA has the proofreading as well as repair and maintenance mechanism for any inaccuracy during the synthesis or copy making a copy of DNA before the cell division. This is the paper yesterday I also showed you, and in this paper, Watson and Crick, they mentioned that the coping mechanism of DNA, and they said they noticed it. If you go through the paper, you can find their statement that you see it has not escaped our notice that the specific pairing was we have postulated immensely suggest a possible coping mechanism for the genetic material. This statement is very powerful, although they had no evidence at that time, but scientists later proved that uh, the ATGC, this type of pairing in the whole DNA is maintained, and DNA has the coping mechanism. And this is the eukaryotic cell cycle, the very simplified illustration. You see there are some checkpoint, for example, S phase, uh, G2 phase, uh, M phase, G0 phase. That means it starts from here, G0 phase, G1 phase, S phase, G2 phase, and then mitosis taken place. And mitosis is the process of making one cell into two. So in the quiescent or G0 phase, then G1 phase growth, phase one cell become enough growth and prepare for uh, synthesis of the DNA at the S phase, starting of the DNA synthesis. And then G2 phase, growth phase two, it is another distinct phase. And after the DNA synthesis, the cell is ready for mitosis to divide into two cells. So eukaryotic cell synthesis is taken place like this way. So as I mentioned, the general principles must be ready. Cell must be ready at G1 stage. Must all start at the same time, G1 to S phase, and must know where to start. That is origin of replication. And then finish, that means must all finish complete S phase, and must ensure that each piece of DNA is replicated only once at this stage, proofreading as well as repair and maintenance and taken place. Therefore, must know where to finish. That means then complete the DNA replication at the S phase, and then it moves to the mitosis to make a daughter cell and transfer the same DNA to the daughter cell. So in replication accuracy or fidelity is a big issue because proofreading is taken place at G2 phase. After S phase, when DNA is synthesized in G2 phase, proofreading or repair and maintenance is taken place. So must distinguish between original and copy. Epigenetics. So epigenetics, I am not sure you heard about it or not. No difference at the nucleotide level, ATGC, but in the side chain, there might be methylation and other in the sugar or other structure can be taken place. So epigenetic change can be also taken place during DNA replication. So DNA replication summary is occurs during S phase of the cell cycle, semi-conservative. It is called semi-conservative, not 100% conservative. And it happens from 5' to 3' direction, and leading strand, lagging strand, and RNA primer. These are the important point about the DNA replication. So single origin in bacteria, multiple origin in eukaryotes, bidirectional, that means 5' to 3', and autonomously replicating sequence elements from each cloned origin. So there are a little difference between prokaryote and eukaryote, the origin of the, uh, DNA replication. Because in case of eukaryote, you see there are mitosis and meiosis, but in case of bacteria, binary fission is taken place. DNA replication, how it takes place, you can see it is semi-conservative. It can unzip like this way, and then template strands can fill up the complementary single strand and make it double strand. So replica strands are taken place like this way, and from one parent duplex or a double-stranded DNA, result two daughter duplex double-stranded DNA. DNA polymerase is involved in DNA synthesis. Always occurs by adding nucleotide to the 3' to OH of the growing strand. You see this here, this growing strand, and it is taken place from 5' to 3'. Synthesis is always in the 5' to 3' direction. You must need to memorize. And this is another image, simplified image, how DNA replication is taken place. Like I previously showed the 3D structure. This is a planar structure. You can see unzipped, and then how it is, primers are filling the gaps to make the double strand. So leading strand from 5' to 3', lagging strand is going on. So this is similarly, so polymerase enzyme is involved because DNA is a polymer, and gyrase enzyme, another enzyme, is also involved in the uh, DNA replication process. So replication enzymes, DNA polymerase matches the correct nucleotides, then joins adjacent nucleotides to each other when DNA is unzipped. So this is one of the very important enzyme involved in DNA replication. Another enzyme is involved in DNA replication is primase. provides an RNA primer to start polymerization, and ligase joins adjacent DNA strands together, so fixes the nicks. So ligase is always, just pasting and making the intact double-stranded DNA when a polymerase enzyme finds the correct nucleotide and add it to the single strand to make the double strand. Ligase is the pasting uh, uh, the real smoothly and joins adjacent DNA strands together. Another enzyme unwinds the DNA and melts it. During DNA replication, I told you that a double strand must be unwind, or how to say, unzip. So helicase is here involved in doing that job. Single strand binding proteins also involved. So lots of molecular regulators are involved in DNA replication mechanism. Keep the DNA single stranded after it has been melted by helicase. When helicase melted it and it divides, on a zip, and then a single strand binding protein helps to, keep the DNA single stranded after it has been melted by helicase. Gyrase enzyme, an enzyme within the class of topoisomerase, and is a subclass of type 2 topoisomerases that reduces topological strain in an ATP-dependent manner while double-stranded DNA is being unwound by elongation RNA polymerase. So several enzymes you understood they are involved in DNA uh, uh, replication. Another enzyme, telomerase, finishes off the ends of DNA strands, from one double strand to uh, uh, one double-stranded DNA to two daughter double-stranded DNA. So lots of enzymes, lots of molecular switches are involved. So day after day, our DNA is damaged by radiation. It is now a fact and well understood. UV radiation, free radicals. Free radicals, during metabolic process when you are in stressful situation, lots of free radicals, right? Singlet oxygen, hydrogen peroxide is developed in the cell, and they are also, problematic. They are called reactive oxygen species, can damage the DNA. And other carcinogens, other carcinogens for through food or other environmental toxins, they are carcinogen. For example, arsenic, or even some a drug, or some other chemical substances. Carcinogen means they can cause damage in the DNA. But despite that, our DNA at a molecular level is inherent. Inherent means almost intact. Thousands of changes happen in our genome at cellular level, but these changes affect our cells when a process that happens thousands of times a day are divided. The reason why our genetic material is not destroyed by chemical levels is due to the large number of molecular systems that control and repair continuously our DNA. That means our DNA is prone to damage or prone to assault by various factors, including UV radiation, free radicals. Some are, endogenous like free radicals. Some are exogenous like carcinogens or UV radiation. But even though there is a very powerful molecular systems that control and repair continuously our DNA. This is why our DNA maintains its own structure. In 2015, a Nobel Prize in Chemistry was given to three scientists. The Royal Swedish Academy of Sciences has decided to award Thomas Lindahl, Paul Modris, and Aziz Shankar the Nobel Prize in Chemistry 2015 for their mechanistic studies of DNA repair. So today I shall discuss their discoveries. They discovered that how DNA maintains its own structure through a very smart repair and maintenance system. Damage to the genetic material poses a threat to all organisms. To counteract this threat, cells have evolved a series of intricate DNA repair pathways that correct DNA lesions affecting base pairing or structure of the DNA. Today, we understand the molecular mechanism of these pathways in a great detail, in large part due to the pioneering studies of Windal, Modris, and Sankar that opened up the field. They opened up this field because they did lots of work, several decades ago. And now we know in detail, and they were the pioneer in understanding the mechanistic uh, principles of DNA repair mechanism. They are awarded for having mapped and explained how the cell repairs its DNA and safeguards the genetic information, safeguards. And these are the three guys, possibly you knew through the news media in 2015. You may forgot because 5 years ago they got the Nobel Prize. One from Sweden, Stockholm, and Francis Crick Institute, another from Duke University, USA, Paul Modris, Lindal from the Francis Crick Institute, UK, and Aziz Shankar. He is originally a Turkish, but when he received the Nobel Prize, he was at University of North Carolina, USA. So, what is the background of their study? The human genome encodes the information needed to create a complete human being. During every cell division, more than 3 billion DNA base pairs are replicated. Our DNA contains about 3 billion base pairs. Through human genome sequencing, we understood this information. And copies of the genome are transferred to the daughter cell. The DNA replication machinery responsible for this task still makes occasional mistakes. This is very important point you should note down. During replication process in the cell, without carcinogen or UV, even occasionally DNA replication machinery responsible for this task still makes occasional mistakes. Given the size of the human genome and the large number of cells in a human body contains about 3.7 into 10 to the power 13 cells. So sometimes makes mistakes which will inevitably accumulate during the lifetime of an individual. If a mistake is taken place, then it accumulate the mistake, and some of the mistakes are very dangerous. So most of these errors naturally happening in the cell during replication, the errors in copying of the DNA will remain silent. Most of the errors, no problem, but they can also cause serious diseases like cancer and others. If a certain mistake in a certain gene, which is very essential for the health or well-being of the human, then the consequences is severe. Despite its essential role in storing genetic information, the DNA molecule has limited chemical stability and is subject to spontaneously decay. Processes such as hydrolysis and oxidation occurs at significant levels in vivo, in part due to reactive metabolites continuously generated in various physiological processes that I mentioned, reactive oxygen species like singlet oxygen or O3 or hydrogen peroxide. They are called reactive oxygen species, and they are generated through the physiological process, especially when you are in a stressful situation. Either it is biotic and abiotic or abiotic stress, reactive oxygen species are generated at higher rate. But there is a system in the cell also to neutralize those charged uh, radicals. And if neutralization mechanism fails to neutralize them, then they can attack the DNA molecule and can cause decay. In addition, external factors like radiation, genotoxic chemicals, that is carcinogens, will further stimulate of DNA damage formation. The inherent instability of DNA constitutes both an opportunity and the threat. DNA lesion can block important cellular processes such as DNA replication, transcription, and cause genome instability and impair gene expression. Lesion can also be mutagenic and change the coding capacity of the genome, which can lead to devastating diseases and conditions associated with genome instability, including cancer, neurodegenerative disorders, or biological aging. Biological aging means you can senescence can be proceed very faster. So people at the age of 30 maybe look like at the age of 70. So these unexpected happenings can be how to say triggered by the wrong or wrong repairing mechanism or lesions in the DNA sequence. That means DNA lesion. DNA lesion means as in the DNA sequence as something wrong sequence or wrong pairing of the nucleotides as well as bases. So furthermore, mutagenic chemicals, there are lots of mutagenic chemicals, for example, EMS, ethyl methyl sulfonate is available. There are colchicine also mutagenic, lots of mutagenic materials available in nature and also synthetic. And radiation, can also have a healing effect. They can, for instance, be used to treat cancer by introducing DNA lesions that halt cell proliferation and stimulate programmed cell death. I told you it is also an opportunity, the inherent instability of DNA, because a cancer patient sometimes doctor suggests to do radiation therapy or chemotherapy. And in both cases, those chemicals or radiation can damage the DNA of the cancer cell and cause lesion so that cancer cell cannot proliferate and it moves to the programmed cell death, because cancer cell has some ability to avoid programmed cell death. That means every cell in our body or they die after certain period of time. And it is called programmed cell death, apoptosis. But in case of cancer cell, they never die because they have the mutation in the DNA. And if you can, use the radiation or some other substances to make a lesion in the cancer cell DNA, then you can stimulate programmed cell death, that is apoptosis, or can stop the proliferation of the cancer cell, because cancer cell always proliferate. But in case of other cell, depending on the tissue and organ, and they are pre-programmed how many cells will be there, as well as their division is controlled. But in case of cancer cell, they have the uncontrolled division. This is why tumor is developed, and they sometimes migrate from the origin to get the new space and nutrition, which is called metastasis. Metastasis means movement of the cancer cell from the origin to the other places, and it is really deadly, and it is difficult and last stage of the cancer and difficult to control. The cell has developed ways to counteract DNA lesions and to keep DNA mutations at a tolerable level. Mutation means lesions, something, some deletion or rearrangement, or some change in the DNA sequence. A number of different DNA repair mechanisms correct lesions and safeguard the integrity of the genome. Four fundamental DNA repair pathways delineated by Nobel Prize laureates will be discussed in this lecture. First is we need to summarize the causes of DNA damage. Chemical mutagens, radiation, free radicals, they are the major causes of DNA damage or having a lesion in the DNA. Radiation, generates the energy deposition in the DNA, and DNA damage taken place. Here you can see how radiation can, cause DNA damage. Here you see it can affect water. Radiation can hydrolyze, radiolysis of the water. You see some electron is released from the water, and hydroxyl group can affect the DNA molecule, the backbone, and can cause hydrolysis, and it is a mutation in the sequence, and it is the lesion. And similarly, respiration and aging, carbohydrate and oxygen during respiration, energy also generate, and sometimes I told you superoxide, hydrogen peroxide, and these are the called reactive oxygen species. They are also highly reactive to the DNA, like which you have seen in case of radiation. They can they are reactive and can cause hydrolysis and other ways that damage the DNA. So DNA repair, despite 1,000s of alterations that occur, thousands of alterations that occur in DNA each day, few are retained as mutation. That means in our body, thousands of alterations are taken place, alterations in the DNA, that means DNA lesion taken place, but a few retained as mutation, but most of them are repaired. Efficient repair mechanism are exist. This is why uh, repairing is taken place. Importance of DNA repair highlighted by number of genes devoted to DNA repair, mutation rates with inactivation or loss of DNA repair gene, and these are the important points. And defects in DNA repair associated with several disease states that I mentioned. One of the important is the cancer. So cancer is nothing but uh, uh, a mutation in the DNA specific gene, and which is not repairable by the natural system of DNA repair and maintenance mechanism. So DNA replication and repair disorders can result several diseases. For example, Fanconi's anemia, hereditary non-polyposis, colon cancer, or Werner syndrome and geroderma pigmentation. But if you see the frequency, in case of Fanconi's anemia, among 22,000 population, only one. And in case of hereditary and non-polyposis colon cancer, out of 200 patients, one, so this rate is too high. Deficient mismatch repair, deficient excision repair. So these are the deficiencies, defects, or deficient helicase enzyme, deficient excision repair, geroderma pigmentation, and Warner syndrome. I shall show you maybe some of the figure. Life exists, so DNA must be repairable. How stable is DNA really? Thomas Lindahl started wondering towards the end of 1960s. You can understand. Thomas Lindahl started this work. He posed this question: How stable is DNA really? In 1960s. In 1960s, people that thought that DNA is 100% stable. But he was suspicious. He had a out of the box thinking, and he posed the question for his research. At that time, the scientific community believed that the DNA molecule, the foundation of all life, was extremely resilient. That means stable. Anything else was simply out of the question. So at that time, people didn't ask such type of question. But you see, Thomas Lindahl made such a question. Uh, that you can call out of the box question. Evolution does require mutations, but only a limited number per generation. So if genetic information were too unstable, no multicellular organism would exist. This is very, very important question because people thought DNA is stable. Their logic was if genetic information were too unstable, no multicellular organism would exist. During his postdoc, Lindahl, at Princeton University, USA, he worked on the RNA molecule, a molecular cousin of DNA, because RNA is single stranded and it is easy to handle. He didn't start his research on DNA molecule. And RNA is very easy to make a decay because it is less stable than DNA. It did not go well, but when he did his postdoc, it did not go well. In his experiment, he had to heat RNA, but this inevitably led to the molecule's rapid degradation because if you offer heat to RNA, then immediately degrade. Coronavirus vaccine, most of them are mRNA. This is why it is suggested to keep minus 80 degrees Celsius or like that. RNA is very unstable. At room temperature, if you isolate the RNA, you cannot keep it for longer time and very rapidly degrade. So he didn't manage the RNA molecule to study whether a decay is taken place by some other factors. It was well known that RNA was more sensitive than DNA. But if RNA was destroyed so quickly when subjected to heat, could DNA molecules really be stable for a lifetime? This question took hold in Lindahl's mind. Although he failed in his experiment, but he thought a cousin of DNA molecule, RNA, if it is too unstable under heat, then how could DNA molecules really be stable for lifetime? He began to look for an answer to that question, and some straightforward experiments proved that his suspicions were correct. DNA underwent a slow but noticeable decay. He did lots of experiments, and finally he found that DNA underwent a slow but noticeable decay. Lindahl estimated that there were thousands of potentially devastating injuries to the genome every day, a frequency that was clearly incompatible with human existence on the earth. His conclusion was that there must be molecular systems for repairing all these DNA defects. And with this idea, Thomas Lindahl opened the door on an entirely new field of research, that is DNA repair and maintenance system. So special enzymes remove damage in DNA. It was his, experimental discovery. He worked with not human. You see, he worked with bacteria. Bacterial DNA, which just like human DNA, consists of nucleotides with the bases adenine, guanine, cytosine, and thymine. One chemical weakness in DNA is that cytosine easily loses an amino group, which can lead to the alteration of the genetic information. In DNA as double helix, cytosine always pairs with guanine, but when the amino group disappears, the damaged remains tend to pair with adenine. So you can see how lesion is taken place. Therefore, if this defect is allowed to persist, a mutation will occur the next time DNA is replicated. Lindahl realized that the cell must have some protection against this and was able to identify a bacterial enzyme that removes damaged remains of cytosine from the DNA. Very interesting findings. And he found that, yes, he found that in the DNA, you see new DNA strand when DNA is replication. When the cell divide, all the chromosomes are copied. The DNA replication machinery unwinds and DNA helix and forms two new strands. You see these new strands they form because it is unzipped, and 5 prime to 3 prime it is moving on, and adenine pairs with thymine and guanine with cytosine. So this is the common. But if cytosine loses some amino group, then it can make a different story. So base excision repair, single base, if somehow wrongly placed or pairs, then there must be some mechanism to repair. Bit by bit, Lindahl pieced together a molecular image of how base excision repair functions. A base excision repair also occurs in human beings, and Lindahl managed to recreate the human repair process in vitro. And the decisive factor for Thomas Lindahl was the realization that DNA inevitably undergoes change, even when the molecule is located in the cell's protective environment, that is, in the nucleus. And however, it had long been known that DNA can be damaged by environmental assaults such as UV radiation. So Lindahl found that not only UV, even endogenously, DNA can undergo changes, undergoes change, and he proved in bacteria. Base excision repair. He found first a simple base, maybe wrongly, how to say, pair with another base, and in that case there is a surgery, that is repair, taken place during the cell division process. And here I can show you very nicely how he showed. For example, guanine should bind with cytosine, but here you see uracil. So this is a wrong uh, uh, pairing, and during DNA replication it takes place, or some other reasons. In that case, you see DNA glycosylase. DNA glycosylase is an enzyme which easily find where is a specific base, wrongly uh, uh, wrongly paired. So identified this one uracil, and then epi side you see remove the base, leaving deoxyribose sugar, and it is removed from here, and thereby producing an abasic or a purinic or epimidinic side by base flipping out. This flipping out is very important. Epi endonucleus then cut this, phosphate and sugar base frame, and cut it and remove it. Deoxyribose phosphodiesterase epiendonuclease cuts phosphodiesterase backbone. This is phosphodiesterase backbone, phosphorus and sugar backbone, deoxyribose sugar and phosphate group. So it is cut here like a surgeon. So and remove the uracil, and then synthesize the cytosine and place it. You see, DNA polymerase replaces the missing nucleotide. So wrong one removed, and then DNA polymerase replaces cytosine here, and then ligase enzyme pasted the deoxyribose and phosphate diesterase, and it is repaired. So the DNA ligase seals the nick. Nick means this cut. This is the very simple presentation of the discovery of Thomas Lindell, how if a single base is wrongly paired, uh, then DNA has the mechanism to identify it and remove it like a surgeon, and single base repair mechanism can be taken place. So this is very interesting discovery, and it is happening always in the living eukaryotic organisms. Otherwise, lots of similar mistakes can, collapse the inherent structure of the DNA, and obviously affect the species species identity from generation to generation. So this is all about today's lecture. Here is the in detail of base excision repair. Here you can see uracil is wrongly instead of cytosine. Uracil is bind with the guanine, because cytosine can easily lose an amino group and forming a base called uracil. If amino group is released, then it become uracil. And uracil cannot form a base pair with guanine. This is the big problem. In that case, you see an enzyme glycosylase discovers the defect and excise the base of uracil. And then another couple of enzymes remove the rest of the nucleotide from the DNA strand. Couple of enzymes you have seen in the previous slide. And then DNA polymerase fills the gap, and ligase, DNA ligase sealed the gap, and it is repaired. So how base excision repair is taken place, the mechanism you have seen, and it is universally happening. So this is all about today's lecture. Next lecture we shall discuss some more topic, that is, if a nucleotide is wrongly lesion is taken at nucleotide level, how it is repaired, and how it is maintained. Uh, and thank you very much. So you can ask any question. Have you heard about this topic before in your any lecture? How base excision is taken place, the whole mechanism, or DNA repair and maintenance system in your previous any course or lectures?
No, sir, I did not have.
So it is you for the first time for you. How about Shafi?
It's also new for me, and
Is it the first time you have learned this new information?
yes, sir. It's also new for me.
Okay, Shafi, your microphone needs to be corrected because we cannot hear you very clearly.
Okay, sir.
My question was, have you received a similar lecture before in your undergrad or master's level?
Uh, no, sir. In this point of view, I don't have this lecture.
Shafi, we cannot hear you clearly. Can you check your microphone?
Hello. Hello.
Yes, I can see you now. But have you received a similar or similar topic in any previous lectures you attended during your master's or undergrad?
No, sir. I do not have.
Okay, then it is very good because this is very, very important when you go for genetic engineering or do any biotechnology. This fundamental understanding we included in this course so that you can deeply understand how DNA repair and maintenance is taken place. You see, DNA has its own mechanism to repair a wrong base and even later I shall tell you, even a nucleotide, wrong nucleotide or a lesion, maybe a damage in the DNA sequence, if somehow happen, it has the mechanism to repair. And this is an inherent principle in the gene editing by CRISPR-Cas method. This principle is very valuable. If you can cut the DNA in any place by using CRISPR-Cas system, and immediately DNA repairs. And during repairing process, it loses some of the nucleotide. As a result, if you target a specific gene and want to make it non-functional, you can successfully do it, and it is called gene editing. If DNA has no such repairing and maintaining system, we cannot do it. And the knowledge of DNA repair and maintenance is very fundamental for understanding the gene expression as well as the regulation and gene editing and other engineering activities that we are doing in biotechnology. So do you have any question? And is it clear, the lecture?
Yeah, it's clear. And Linda's experiment is really interesting.
Right. So, what is my suggestion? Please visit the Lindahl's website. You can find him in Wikipedia or Nobelprize.org, both of you, and try to understand why he did why he received the Nobel Prize in physiology or medicine, and to know the person and his journey to discover this, fundamental information about the DNA repair and maintenance mechanism. Uh, it would be very nice if you go through his biography as well as his journey of unknowns, and he made really big breakthrough and opened a new area of science. Moina, do you have any question?
No, sir.
You understood my lecture?
Sir.
Great. So, Moina and Shafi, please go through the website, browse the website to know more about DNA repair, base excision repair mechanism. You can find even YouTube, a link also on this topic, and get more insight because lots of research have been done on this topic. I tried to tell you the story in a simplified manner, and hopefully next lecture we shall discuss if big lesions taken place at nucleotide level, how DNA repair and maintenance mechanism can handle it. And Aziz Sinker and Paul Modris, these scientists, they did lots of work and discover all those stories. Thank you very much.